track data on who reviews the recordings and what their grades are. How many of you guys actually use those? Oh, wow. Okay. I did not know. Okay, so the very last connective tissue doesn't seem like a connective tissue at all. It's blood. So see if anybody can remember why this tissue that doesn't seem at all like a connective tissue is actually categorized as one. Go ahead. That's not the reason. It's good logic, though. Go ahead. Yeah, it's derived from mesenchyme. So basically, its adult category is really derived from its embryonic origins, right? So if it seems counterintuitive that it's a connective tissue, yeah, it is counterintuitive that it's a connective tissue. But it came from a connective tissue. It's actually a connective tissue made by a connective tissue because it's made by bone marrow, right? Okay, so we're just going to do a quick version of blood. You can learn a lot more detail about blood, and we will in the future. But blood is a connective tissue, but the matrix is almost completely water. The matrix is called plasma. It's about 90% water. It's got some proteins and stuff in it as well. But the matrix is called plasma. And then, of course, it has cells in the matrix. Although in blood, FYI, they don't say blood cells. They call them formed elements, and here's why. One of them isn't actually a cell. So let's go through them, and then I'll point out what that's all about. So if you look at blood just generally, it has 3 categories of formed elements in it. The most abundant thing in any blood smear that you're going to see is going to be erythrocytes, which are what? Red blood cells. And they don't have a nucleus when they're fully mature. And they have this really regular shape called a biconcave disc, right? It's like a donut without the hole pushed all the way out, right? Super duper regular in size and shape and very, very abundant. In males, they have about, in a big drop, you'd have about 4 to 6 million red blood cells per big drop. And in females, you have almost 1 million less because testosterone causes erythropoiesis. It causes red blood cell production. What's the primary function of red blood cells? Transport gases. Mostly oxygen, but also a little bit of carbon dioxide. Okay, and then you've got your white blood cells or leukocytes. White blood cells, how do you identify them just for right now? Anything bigger than a red blood cell with a nucleus is a white blood cell, but they don't all look the same. They have really different sizes and variable shaped nuclei. Here's a couple of different examples. These are also white blood cells, and this is one. They're leukocytes. They have varied functions, but let's simplify the function. Defense and immunity. Defense means anytime I run into a pathogen, I try to take it out, right? But immunity is remembering who attacked me the last time and being ready to take them out, yeah? So if, for instance, you walk around a corner, somebody clocks you in the face, you just defend yourself. You walk around the same corner, you got a baseball bat, right? You're like, that's immunity. And immunity, by the way, is part of what early exposure to pathogens and or vaccinating does for you. FYI, get your flu shot. The flu is terrible and has already closed down 2 schools in Utah this year, okay? And it's coming from south to north. It killed a bunch of people in Australia. And by the way, even though the flu shot, FYI, is not outrageously effective at protecting you from getting the flu, one of the things that is really effective is removing the likelihood of spreading it, which is really, really important for the elderly and for children and for people with depressed immune systems. So you're not getting vaccinations just for you. Okay, so leukocytes. And then last but not least, these are called thrombocytes, but they're actually not cytes, FYI. Yeah, they're platelets. What's the primary function of a platelet? Yeah, clotting. One of the million things that's involved in clotting, but it's one of them. Now, why are they not whole cells? They actually are from whole cells, these big megakaryocytes that make a whole bunch of clotting factors. And they come from your bone marrow, just like the rest of your blood-formed elements. But then they burst, and they can't go through cell division. They're just like a little bag of clotting factors, really. So that's why instead of calling all of these collectively blood cells, they call them formed elements. It means solid things that might be cells inside your blood. Good? Okay. So we do blood in more detail in later chapters. Go ahead.
On the test, do you want us to call them thrombocytes, or do you want us to call them platelets?
I don't really care. Would you just know both terms and I will accept either term? Okay, because they're both really commonly used. So it would kind of be silly to not know both of them. Okay, so now let's do the same kind of study tips that that we did with epithelial tissue. Let's do them with connective tissue. And connective tissue is a little bit more complex. So let's talk about the kinds of questions that you could ask with any connective tissue. You ready? So let's start with this one. Okay? That's not the question, though. So here's the deal. Every time you pop up a connective tissue, what people immediately do is answer one question. But here are all the questions I could ask about any connective tissue. So instead of the question you expect, I'm going to ask a different question. I'm going to ask, what category of connective tissue is this? Do you remember the categories?
Connective tissue proper.
Proper. What was the other one?
Dense.
No. Proper. No. Proper. Supporting fluid. Okay, so she got it. It's proper. Okay? Then I'm going to ask you some other questions that you might not expect, but are totally fair questions. You just need to condition your brain to look for these. What's the name of the cell?
Fibroblast.
Fibroblast. Okay. Now I'm going to ask you a question that you might have expected. Of the propers, which category does this fit into? Dense. Good. Okay. And then I'm going to ask you, what is this? What kind of fiber? Collagen fiber. Does that make sense? And then the question that everybody hates and does really poorly on is specific body location.
Tendons.
Tendons. Good. Okay, you got it? So what you need to do is you need to look at it and you need to go past what the C anatomy student will do, which is just naming the tissue, and get to what the A anatomy student will do, is all of the questions that you could possibly expect. Does that make sense? So let's do some more. You ready? Okay. Name this structure. Lacuna. Right, exactly. That makes sense? Okay. Now tell me what general category of connective tissue this is.
Cartilage.
It's not one of the choices. Supporting. Yes. Now tell me a body location for this tissue. Nope, the nose is the other one. The ear, external ear, right? So, and now tell me what this fiber is. Elastic fibers. And now tell me what tissue it is. Elastic cartilage. Okay? So there are lots of fair questions. You just have to be able to organize with every time you look at it to know which questions you're talking about. Okay, so let's do this one. Is this a loose or a dense connective tissue proper? Loose. Okay. What am I pointing to right here? A lipid vacuole. So how would I get you to identify the cell? Well, because it's my job to write a clear question, I would have to say, name this cell. Yeah? Okay. So I have to be really clear about what I'm asking for, or else I'm going to get a hodgepodge of answers, right? Does that make sense? Okay. So what general category of connective tissue is this? Connective tissue proper. Name a specific body location. Surrounding most organs. Where else? Subcutaneous layer. People do this all the time. Why is this wrong? Stomach. Well, first of all, it doesn't. The stomach's an organ. It doesn't have any adipose on it, right? What they were really trying to say was the subcutaneous layer of the abdomen, which is right. But choose something easier if you can't come up with subcutaneous layer of the abdomen. There's a lot of things that are true. A lot of times people write, your butt. And I'm like, talk about your own damn butt, dude. So let's do this one. Name the category of connective tissue. What is this?
Plasma.
Good. Now, what is this?
Ground substance.
Ground substance. What is this fiber?
Elastic.
Elastic fiber. Name me a body location for this tissue. Around most organs, under mucous membranes, right? So again, with body locations, unless it's one that I always talk about in class, like let's do one last one. What body location have I given you for this? Intervertebral disc, pubic symphysis. So Meniscus of the knee, exactly. If it's one that I've talked about repeatedly in class, I expect that you've heard me talk about it. Other than that, I don't care which one you write down, right? Does that make sense? So what is this fiber?
Collagen.
Collagen, even though it doesn't look like it. Yes? So how do you get good at this? What you do is you pull out your histology slides and then you run through this list until you're confident with multiple iterations of the same slide, getting all of these questions right. And then you're going to be a histology badass, right? You are not going to flunk histology. That makes sense? Because I can't think of any other questions, otherwise I would have written them down. Okay? So it's just your job to sort of be really rigid about and disciplined about your studying with histology, and then you'll be fine. Okay? All right, so nervous tissue is really easy. Actually, it's not really easy. The way that they teach nervous tissue in this chapter is really easy. But nervous tissue, let's talk about its functionality, and then we'll talk about its structure, and then we'll talk about what its structure is really like, even though you won't have to look at that histology. So functionality of the nervous system is basically threefold. Nervous tissue is information in, information processed, and information out really quickly, too. So a really simple example is sticking your hand on a hot stove, right? And so what I have to have is information in, which is sensation, and that goes through the nervous system, okay? Information in, but then I have to process the information. And what does the processing look like? Well, with this, it's all subconscious, but it would go hot, right? And then how hot, right? Too damn hot, comparing it to your ideal, right? And then it will say, this is the response that we need, and send it out, right? So sensation, integration, response, yeah? And response is stimulate skeletal muscles so that you move it, right? So that's a really simple one. But sensation, integration, and response isn't always really, really simple, right? It could be, it's freaking cold, right? Think about what happens when I'm cold. Well, I go shiver and I go put on a jacket because I'm cold, right? Or it can be super complex, right? Like information in about political candidates. Oh crap, information processing about how I want the world to be. Information out and push the button. I'm pretty sure that mine is not hooked up in Utah. I don't know what the hell's going on. But anyway, so it's not always a really simple pathway, but it's always going to be nervous system loop. Does that make sense? Okay, so of course you find nervous tissue in the brain, the spinal cord, the nerves, the sense organs. We're going to talk about it in excruciating detail this semester because nervous system is my favorite, second only probably to reproductive system. And then let's talk about the types of cells that you run into with the nervous system. So if you look at the nervous system, this is the picture that they show you. This is from a specific part of the brain. It's not from the spinal cord, and it looks different in different parts of the brain. It looks different in the spinal cord. It looks different in the nerves. So this is just like one little nervous system tissue slide. So the only reason I'm telling you that is that if you go further into the nervous system, you are going to see a hell of a lot more than this, right? But for right now, let's just keep it relatively simple. The nervous system is composed primarily of two categories of cells. There are these big star-shaped cells called neurons. Yeah, and we'll talk about those in a little bit of detail. And then there are all of these other more abundant cells, but much smaller cells. And those are either called neuroglia or glia. Either one's just fine. They called them glia because when they found them, they thought they were just glue, which is what glia means, just space fillers between the neurons, because everybody knew that the neurons were the stars of the show. We will see that they were totally wrong about glial functions. So let's talk briefly about these kinds of cells. So the neurons are the electrically active cells that send electrical signals like 250 miles an hour and release neurotransmitters and communicate with each other and with muscle cells. They're the stars. They're the reasons that you have your personality and your intelligence and all of that kind of stuff, and they get a lot of attention. Super, super interesting. Now, if you look at them, what they have is a cell body with a bunch of processes sticking off. You can't tell that this process is different than the others in that picture, but you could if you did a drawing, right? So if we do a drawing, you can see that this particular type of neuron, which is called a multipolar neuron, what it has is a bunch of short processes and then one long one, right? But do you see that it's kind of arbitrary that they called that one the axon? Because you can't see that in the histology slide. right? I can't expect you to know which one's an axon from a picture like this. But if you have a drawing, you can tell. So as functionally, this is called the soma or cell body. It's where almost all the organelles are, okay? And then these are basically receivers. What they are is they're called dendrites, and they receive information in. They have a whole bunch of cell membrane receptors on them. So they're sensitive to a bunch of different inputs. You guys with me? So they receive information, and then the axon's job is to send information away from the cell body. So information comes in and goes out. So the good part about that is just with a little anatomical knowledge, you can look at a neuron and tell the direction of information flow instantaneously, right? Right? And then if you put 5 neurons in series, you could tell which direction information was flowing through all of them, right? That ends up being really useful in physiology, is just to have that practical snapshot anatomy understanding. Okay, so the neuron is going to get all of the attention. It's eventually going to release neurotransmitters from this side. And we think about neurons a ton, and we know about 30 different neurotransmitters. Some of them make you smart, some of them make you stupid, some of them make you addicted, some of them make you crazy, depending on proportions. And I talk about that stuff a lot in physiology. I love neurophysiology. But for right now, let's talk about the support staff. The support staff of the neuron. They are way more abundant. They make up half of the brain volume, even though they're not as big. And they are collectively called glial cells. There's actually 6 different kinds of them, but you can't tell the difference right now, so we'll learn the 6 different kinds later, okay? But they don't do nothing. So the metaphor that I use for neurons and glial cells is basically the neurons get all the attention. So imagine that these two have been movie stars since they are children, and they had to do one thing extremely well. And in order to do the rest of their life functions, they need someone to, for instance, tell them which side of the car the gas goes in, right? How to pay their bills, what kind of coffee they like. They need a bouncer or support staff or security staff. So these are basically like the Olsen twins. And this is all the people that are supporting them. And what happens to the Olsen twins when their support staff bugs out?
Drugs.
And yeah. Like, I cannot function. Probably true. Yeah. Okay, so what do these guys do? What kinds of things do the glial cells do for your neurons? Well, first of all, they make myelin. Myelin allows it allows the transmission to go from one place to the other. This is basically like a communications director, right? In the absence of myelin, you will start losing functionality. Human physiology, pathophysiology example is multiple sclerosis. With multiple sclerosis, the neurons are functioning just fine, but they lost their capacity to send their message because their communication cells really bugged out on them. Does that make sense? They also are protecting phagocytosing bacteria, because one of the interesting things about the brain is that with the brain and the spinal cord, you have a blood-brain barrier, which keeps nasty stuff from the blood from getting into the brain tissue. It also keeps white blood cells from getting in there, which means since you don't have the cops, you have to have an internal security staff, and that's glial cells. Does that make sense? And then they also nourish your neurons. They know what kind of coffee you like. They know what you want, right? They bring you stuff. They also take out the trash for you. When you make your waste products, they get rid of it. And when they start to go bad, then the neurons start to go bad. For instance, Parkinson's disease is a portion of your brain called the substantia nigra. It's really deep in your brain. And what it's supposed to do is it's supposed to control motor activity, to smooth motor activity. You with me? But with Parkinson's disease, you start losing smooth control of motor activity. So obviously people are like, well, there's something wrong with the neurons, but the neurons look absolutely normal. What's abnormal in that part of the brain is that the glial cells have bugged out on you, and then the neurons can't function properly. Does that make sense? So it's only about the last 30 or 40 years that they've really been looking at glial cell research to figure out what it's affecting neurons. And so we'll talk about glial cells a lot when we get into the nervous system. That makes sense? Okay, so for this, what you would need to be able to do is go, okay, this is a neuron, these are glial cells, right? These are processes. I can't tell which one's an axon versus a dendrite unless I have a drawing, right? That makes sense? And this is the cell body of the neuron. That's about as far as you have to go, okay? Later on, you'll do it in more detail. Okay, so with muscular tissue, we're going to do it a little differently. Since there's 3, and they're relatively easy, we're going to do comparison contrast with the muscular tissues. So one thing about muscular tissue that's kind of weird is that I am going to use a word in a way that it's not usually used in tissues. So the longest muscle cells in the body are up to a foot long. They're so long and so skinny that they are called fibers. But that's not how we use the word fiber in connective tissue. In connective tissue, was a fiber a cell? No, it was made by cells. Does anybody remember what it was?
Fibroblasts.
It's made by fibroblast. And what is the fiber made of? Like, what is it?
Collagen.
Yeah, but what is collagen? It's a protein, exactly, right? So it's a protein that's made by the cell. So this is a little counterintuitive because, of course, now I'm saying a cell is a fiber, but in connective tissue, a cell is a protein. So you're just going to have to get used to it because they use the word fiber for muscle cells forevermore, okay? So when I'm saying fiber, I'm saying cell with muscle, okay? So muscle fibers actually contract. They wiggle things, but they also hold you steady. So they're for postural stability, right? Like I couldn't stand in front of you if I were quadriplegic because I need some level of ab, back, leg, and neck contraction in order to hold myself up. And then sometimes they function, they wiggle, but not to make you go anywhere or to make anything go anywhere, but just to heat up your body. Shivering is fast muscle contraction and relaxation, right? Okay, so what we're gonna do is we're gonna compare the types of muscle tissues, and I'll draw a little sketch. You guys can draw it at the bottom if you want to. Skeletal, smooth, and cardiac muscle, and compare and contrast them. Okay, so first, if you look at the three of them, let's look at the first one. This is skeletal muscle, okay? And skeletal muscle is the longest of the three. Usually you don't see the ends of a skeletal muscle fiber in a histology slide because they're up to a foot long, so you're not going to get the ends of them. If you did see the end, however, skeletal muscle is elongated and it has blunt ends, okay? Smooth muscle, however, is elongated, but it has skinny ends, like little eyeballs or something, like really, really thin eyeballs. So they call it spindle-shaped, but I don't know how many of us have seen a spindle. We're not Rumpelstiltskin, right? So I haven't seen a spindle ever in my life, have you? Okay, so that is smooth. And then cardiac muscle is elongated with blunt ends, but quite often you can see it branch if you look hard enough. So it's elongated. It's nowhere near as long as that one. And then quite often it will branch. Not always, but some of the cells will branch. Do you see this one branching right here, right? And if you look down here, you can see this one going two different directions, right? And then this one going two different directions. Okay, so that's the first thing. The second thing is how many nuclei do they have per cell? So if you look at skeletal muscle is multinucleated, and usually the nuclei are pushed up against the side. So it's multinucleated and they're peripheral. Smooth muscle is uninucleated, and usually they're centrally located in the cell. And then cardiac muscle, uninucleated, usually centrally located as well. So here's cardiac muscle, usually one per cell. Does that make sense? Okay. And then we get to the striations. Okay. So let's talk about the three tissues, striated or not striated. So striations are not the same as fibers. Fibers are the whole cell, and striations are a feature of the cell. Striations run perpendicular to the long axis of the cell. Perpendicular means 90-degree angle. Every semester somebody tells me they don't know what perpendicular means, and it's never a person who doesn't speak English as their first language, right? Which tells me something about the Utah school system. Perpendicular, right? Perpendicular. These are striations. This one doesn't have striations, that's why it's called smooth. This one does. Okay? So let's look at it. Make sure that you can identify the striations. In skeletal muscle, they're really easy to see, right? Super easy, right? In cardiac muscle, they're a little more faint, but you can definitely see them. Do you see them? Okay. This, by the way, is not a striation. It's parallel to the striations, but it's actually called an intercalated disc. And what it is, guys, is where two cardiac muscles are attached to one another, two cells are attached to one another. And they're attached in such a way that they have a really strong association and they can communicate with one another to cause a wave-like contraction. So what types of cell junctions do you expect to be found at the intercalated disc? Gap junctions for communication, and what for strength? Desmosomes. Yeah, and since you have so many of them, it stains really dark, so it looks like a really dark striation. So let me show you a couple of different cardiac muscle. Here's cardiac muscle too, different staining technique with cardiac muscle. Good? Okay. So whenever you see what looks like really dark striations, right, it's usually an indication that you've got, hey, every once in a while it looks like there's a dark striation. That's probably cardiac muscle. Good? Okay. And then branched or unbranched, the only one that will ever branch is cardiac muscle. So look here, right? Watch this branch happen. It's going, and it goes that direction and that direction. Do you see? Okay. This one's going, and it goes two different directions. Not every cell will you see a branch in the slide, but you can see them. The other two don't branch. Okay. And then let's make sure that we can look at smooth muscle. Smooth muscle is the one that people have a little trouble with. They have a little trouble with it often because it tends to look a little like what other tissue? Dense regulars. So let's acknowledge that right now and figure out how to tell the difference between it. Okay. So this is the picture from your textbook, but it usually looks more like this. Okay. This is smooth muscle right over here. This is actually smooth muscle too, but it's just a cross-section of the same tissue. Don't worry about me asking you like that. It just exists that way. But do you see this smooth muscle over here and how easily you could confuse it with dense regular? And now let's acknowledge a couple things. Do you see that this nucleus is inside the cell? Right there, right? Okay, and now let's look back at our, do you see that these nuclei are smashed between the fibers? Do you also see parallel fibers running all the way through and not just what looks like parallel cells, right? Okay. So if you look at it, dense regular really looks like you've got nice smooth hair going between the cells, right? It's running all the way through between the cells. So how do you get used to telling these two apart? You stick them against one another like 25 times until you get it right every time. That make sense? Okay, so this is smooth muscle. So now let's talk about control, nervous system control of these tissues. Which of these tissues are you ever in voluntary control of? Skeletal muscle, and only sometimes. These two you never control voluntarily, meaning that the part of your brain, your cerebral cortex, that has voluntary control of anything is not wired up to your heart, and it's not wired up to your smooth muscle. Can you make yourself secrete something on purpose? No. Can you think about increasing your heart rate and increase your heart rate? No. Can you think it's a really inappropriate time to have diarrhea and just turn it off with your brain? No, you can't. So, FYI, there are ways in which you can train your brain to, your cerebral cortex, to override the part of your brain that does control your heart, but you're never controlling your heart directly. And that takes a hell of a lot of training, a whole bunch of yoga, a whole bunch of martial arts training. You're a freaking CIA operative trying to figure out how to not get caught, right? Most of us cannot do that. We go into a bad job interview and your heart rate goes up and you're just like, oh my God. Or you get anxiety and you're like, is it ever a good time for anxiety? Hell no. But can you control it? No, you're just crazy, just like the rest of us. So you can't control these things. Matter of fact, you can't even control your skeletal muscle all the time. Try to hold your breath to death. That's the diaphragm is a skeletal muscle. Your brain will go, I got this, if you don't do it right. It'll be like, sit down, I gave you a chance, okay? So in addition, one of the really humbling things that occurred to me a few years ago is that really, actually, there's only one single tissue in your entire body that you're ever in control of, and that's skeletal muscle. You are never in control of any of the rest of your tissues in your body. You can't make your connective tissues do anything. You can't make your cardiac muscle do anything. You can't even make your own damn brain cells do what you want, right? Because we would not be in this situation if we could, right? If you'd be like, I definitely want to be an astronaut brain. I need you to make me into an astronaut. And it never did. First of all, I'm too short to be an astronaut. That was really my only impediment, honestly. Not like calculus or physics. Okay, so now let's talk about location in the body. That's easy. Skeletal muscle is called skeletal because it's generally attached to your bones, not always. Smooth muscle is in the wall of any hollow internal organ or tube except the heart. Yeah? And then cardiac muscle is only in the wall of the heart, right? That's the only place that it exists. And now let's talk about speed of contraction and ability to remain contracted, and this is kind of important. Okay, so which of these three is going to be your emergency response tissue that will get you out of the way of an oncoming bus? Skeletal muscle. This needs to be your sprinter. How long can sprinters sprint compared to marathoners? Not very long. They're very quick to respond and they use up their ATP very, very quickly. They cannot maintain contractions for very long, comparatively speaking. So while Volkswagen falls on my baby, I might be able to lift it off on a good day. Can I hold it up? Hell no, you can't hold it up, right? Even if I could lift it off. So which of these three tissues, if the bus chases me for a couple of blocks, am I going to need to bring on second? Cardiac muscle, right? So that one's intermediate in its speed of response. And which one's just sloughing around, not doing a damn thing to help? Dirt, 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 dirt. Smooth muscle, exactly, right? So the speed of contraction is inversely proportional to your capacity to remain contracted, because if you contract really quickly, you burn up your resources really quickly, right? This one can go forever, though, right? And this one's intermediate. That makes sense? So this brings me to what I call the diarrhea race. Eat some super duper duper spicy food. Go to Shannon Thai and order anything above a 3. Have you guys been to Shannon Thai? I had to walk around outside, almost had to buy new underwear. I'm like, oh my God, it's coming out. So the diarrhea race is what happens when your intestines get irritated for whatever reason, and your intestines start to push the stuff out. And they can push forever, and they're just going to continue doing this. And what you're doing is you are pitting a marathoner against a little skeletal muscle sphincter at the bottom, which is a sprinter. And as long as you sprint to the bathroom, you might not have to buy new underwear. However, if you're expecting to do a marathon, it's going to be an uncomfortable social situation, right? Because this is not going to win at the bottom. It is going to come out, right? So it will hold on and hold on and hold on, and then it will just burn out, right? And then it's just going to come down your leg. Does that make sense? So that's the diarrhea race right there. You'll never forget. And then cardiac muscle's just over here going, dudes, what's up? It's like, get it together. Okay, so that is muscle. Let's do a couple little cleanup things about tissue, and then we will move into the skeleton. Okay, a couple things about tissues. First off, what happens to your tissues as you age, and then what about tissue death? Well, tissue death, apoptosis is programmed cell death, right? But when an entire tissue dies, like for instance because of lack of oxygen going to it, maybe frostbite or something like that, it's called necrosis. The problem with leaving necrotic tissue around is that there are pathogens that like to eat dead and dying tissue. And those will cause gangrene. There's dry gangrene and wet gangrene or gas gangrene. And so here's what happens with improper wound care. If you have a wound, of course, there's the likelihood that you can get pathogens in the wound, right? So of course you want to clean it as much as possible, but then in addition, you never want to leave dead and dying tissue around. So if I had a skin flap that was obviously dead, you need to get rid of it. It needs to go away. And then the other thing that's really important for you guys that are going to end up doing healthcare is that wounds have to be cleaned regularly. And they have to be cleaned regularly because there are always potential pathogens. And if you get pathogens in a wound, they can start to, those pathogens that like dead and dying tissue, once they get a hold, they will actually start to kill the tissue ahead of them so that they can make a food source for themselves. And it's called gangrene. Once gangrene sets in, there's very little you can do about it. Quite often, what results after gangrene sets in? Amputation. If you have it on your torso, amputation's going to be a bit problematic, right? So, of course, historically speaking, especially in World War I, and less so in World War II, soldiers got injured, get some shrapnel in somewhere, don't clean it properly, either because you can't, because of the situation that you're in, or you didn't have the resources. And remember that antibiotics are not readily available at this point. And then you have amputation after amputation after amputation. Now, those of you who are not in the military will probably be more likely to run into this dealing with diabetic patients. You are all going to end up dealing with diabetic patients regardless of whether you're going into physical therapy or pharmacy or nursing or med school or PT. It doesn't really matter, or PA, because we have a diabetic epidemic here in the United States. Type 1 diabetes is autoimmune with a genetic predisposition. Not a damn thing you can do about it. Type 2 diabetes is primarily caused by eating and exercise patterns, and pretty much the American eating. Regardless of how you got either one, what happens is you reduce blood flow and depress your immune system, and wound healing becomes really, really problematic. So you get wounds and they don't heal, and then if you don't take care of them, then gangrene can set in, right? You also have poor blood flow to the area, so healing is problematic with diabetes. So a lot of times with diabetics, you will end up with amputations if you don't have proper wound care during that time. In addition, diabetic neuropathy means that if I step on a tack with neuropathy, I don't feel it, so don't care for it until it's later, right? So you end up with situations like Larry H. Miller, who, the car dealership guy around here, who had diabetes and ended up with a double amputation, I think, before he died. I don't know whether he had type 1 or type 2. Does anybody know? He was really heavy, so that didn't help. He had type 1. Yeah. So regardless of which one you get, it's still going to be a wound care problem. Okay, so what else? What happens to those of us who are otherwise healthy but are still going to age? As you age, what happens is your epithelium will thin. So if you just think about surface epithelium for just a second, exfoliation rate does not change as you age, but cell division rate does. So you get thinner and thinner and thinner, right, tissue. To the point if you've ever dealt with elderly people, sometimes their skin is really papery thin, right? So you have to be careful about moving them because you can actually rip skin just by sort of normal care. And then your connective tissue will lose its resiliency because as you get older, your collagen and your elastic break down and they're not regenerated as well. So when you're in your 40s, like I am, you start getting wrinkles, things are heading a little closer to the south, right? So that loses resiliency. And like I said, if you pinch like the skin of an elderly person with permission, it just stays up for a while. And then your muscle and your nervous tissue will also begin to atrophy. But that's not a death sentence. Most of that is due to our activity and eating patterns as we age. As we age, we tend to become less active, both physically and mentally. And it's a use or to lose it with muscle tissue, bone tissue, and nervous tissue. If you are more active, do you know people who are older than you and in better shape than you? Yeah, you do, right? Do you know people who are older than you and more mentally active than you? Yeah, so that's part of how you maintain the activity of this tissue is because if you use it, you actually send more blood flow to it and it doesn't age as readily. Same thing with bone mass. If you don't use your bones, if you're a couch potato, you start losing your bone mass. So a lot of these changes are due to diet, circulation, and activity patterns. And unfortunately, here in the United States, getting older is also more likely to be associated with poverty. It is not true in a lot of westernized countries. For instance, if you go to Denmark, elderly people are not the poorest of people. They are middle class to more. They have socialized medicine. They've got easy access to medicine. So aging looks very different in other westernized countries than it does in the United States. Have you guys ever been to Holland? Have you ever been to Holland? Yeah? Old people riding bikes, 85 years old. Woo! My husband, he's half Dutch, and his aunt, when we went over there last, is 85. She rides her bike everywhere in her little skirt. It's adorable. But she isn't the only one. Everybody, old people are out at the cafes all the damn time. She decided she was going to be mayor when she was 85, right? She's not just sitting around watching Fox News, which we know is bad for your brain, right? So So you do not, this is not a death sentence. I mean, we're all going to die, but leave a good-looking corpse, right? Okay, so how to build an organ. Last thing, well, almost the last thing. We've got one thing after this. So all I want to do here is say, okay, you got a cross-section of an organ, and you're going to look at it. You can have an expectation of what you're going to see. Let's say we're going to do a hollow organ. What do we expect to see on the inside of a hollow organ? What kind of tissue? Epithelial tissue, and then a basement membrane, and then some connective tissue. If it's a wiggly organ, we're going to see what in the wall? Muscle of some sort, right? Then we're going to see some more connective tissue. And then depending on whether it's stuck to something or not stuck to something, if it's stuck to something, just connective tissue on the surface. If it's not stuck to something, it'll have epithelial tissue on the surface, okay? So you can have an expectation. So this is a cross-section of any hollow organ, but let's pretend that we're actually doing the stomach for a second. Okay, so if you imagine the stomach and you look inside surface, touching the lumen, you would see an epithelial tissue. But here's some logic questions for you. If I saw a really thick epithelial tissue, what might that tell you about the function of the organ? Thick epithelial tissue on the inside. It's protective. Yeah, it might be dealing with solids. Yeah, the stomach has relatively thick epithelium on the inner surface, right? However, and if it was thin and had microvilli on the surface, what would that probably tell you? Yeah, intestines, it's doing absorption, right? So what kind of epithelial tissue you run into will give you an immediate idea about the function, right? And that's what I want for you guys. Instead of rote memorization, you're like, oh, I have a hypothesis about this. Under every connective tissue, of course, there is going to be a basement membrane, which is acellular, but it's always going to be there. And then there's going to be connective tissue under there. Let's say the connective tissue could be areolar or dense irregular. If it was dense irregular, what might it tell you about the organ versus areolar? Any idea? What's dense irregular's characteristics? It's really strong in a bunch of different directions. So maybe that would be an organ that was under a lot of tensile stress, right? That makes sense? Okay. And then you're going to move through, the muscle tissue. Anywhere but the heart, it'll be smooth muscle. But this one has one layer of smooth muscle around, right? Circular layer, and then it's got a longitudinal layer. The stomach has an extra layer. So the thicker the smooth muscle, the more wiggling. Maybe the stomach is moving solids, right? So the thickness of the musculature can tell you something. And then on the outer layer, what you are going to have is either connective tissue plus an epithelium, or you just end at connective tissue, right? Does that make sense? The stomach actually has connective tissue plus an epithelium because it's like hanging out by itself, right? Okay, so now last tidbit is body membrane. So now we're moving from histology to organs, but we're going to move to something that doesn't seem like an organ. Okay, so a body membrane is 2 layers of tissue at least, okay? And if you are more than one layer of tissue or more than one kind of tissue functioning together, aren't you an organ? Yeah? Even if it doesn't seem like you're an organ. So let's talk about a mucous membrane. A mucous membrane is basically an organ that is covering surfaces that are moist and exposed to the outside of the body, okay? Moist surfaces exposed to the outside of the body. So your entire digestive tract all the way down, right? Your entire urogenital tract and your entire respiratory tract are all covered with an organ called mucous membrane. Does that make sense? So a mucous membrane, really, really clinically important, because obviously in order for something to get inside the body, it's quite often going to have to cross a mucous membrane to get inside the body, right? So that means that they're set up for transport, which makes them very, very susceptible to pathogens. Aren't those the body systems that are most likely to get infections? Digestive, respiratory, and urogenital? Yeah? So why don't we just close them off? Well, because you die. You actually need to bring things in. So what do? Well, we have to live with the possibility of getting pathogens, yeah? And then have a protection system for when you do get pathogens, right? That makes sense? So let's talk about what kinds of tissues make up mucous membranes. Well, they're always going to have an epithelium, basement membrane, and then a connective tissue. The connective tissue is almost always areolar underneath there, but the epithelial tissue depends on where it is. So let's think about just the digestive system for a second. The proximal and distal ends of the digestive system are going to be what kind of epithelial tissue? Do you remember?
It's non-keratinized.
Non-keratinized. Stratified squamous. Okay, good. That's proximal and distal. But then as we start moving through, we're going to change tissue types. In some places, we're going to have like a thick epithelium in the stomach, but in some places, we're just going to have like simple columnar epithelium with microvilli. So, like I said, as you move through, as the function changes, the structure will change to support it. Does that make sense? So usually the two most common tissues that you find in mucous membranes are either moist stratified squamous epithelium or some kind of columnar, right? Some kind of columnar. Don't know which one until you look, but okay. And then by the way, the combination of these layers, you're going to see this every once in a while, so I'm just going to point this out. This epithelial tissue plus the connective tissue underneath it is often called a mucosa, which is another way of saying a mucous membrane. Okay. Now, last one is a serous membrane. So we met serous membranes before, and we know about their function. Now let's just put together what they're made of. A serous membrane is an organ. So you have an organ on top of your stomach because it's two layers of tissue, right? The superficial layer is a modified simple squamous epithelium, modified because it actually has secretory capacity. It produces that serous fluid, right? And then it's got a basement membrane, and then it's got areolar connective tissue underneath it. Does that make sense? Okay. So how can you avoid abrasion if you have two layers of just simple squamous epithelium? Because they're both secreting a lubricating fluid, right? So they're both lubricated. Go ahead.
So in all of these cells, would you find the goblet cell? Because that's what is produced.
You can find them. They're a little difficult. That's one of the reasons that we didn't have you learn the histology of a serous membrane. But yes, it actually has secretory capacity. And the secretory capacity in serous membranes is mostly in the epithelial tissue, but some of it actually comes up from the connective tissue as well. So it's a hard slide to look at. We don't usually show them to you. But yes, good question. Okay, so by the way, this word mesothelium, there's a bunch of different kinds of simple squamous epithelium. You've got like endothelium and mesothelium. I never test you over those. I'm just going to say it's simple squamous epithelium. But FYI, you're going to run into those in the future. And really, it's just like this kind of simple squamous epithelium versus this kind, right? Still simple squamous epithelium. And then, of course, the last membrane is your cutaneous membrane, which is just a fancy way of saying your skin. But I want you to see if you can tell me what cutaneous membrane is made of without looking, okay? The cutaneous membrane or the skin. The superficial layer is made of what kind of tissue? Keratinized, stratified, squamous epithelium. And then a basement membrane. And then does anybody remember what's underneath? Areolar and? There's two. And dense irregular. Yeah? Because you got the papillary layer and the reticular layer of the dermis. And so you got a combination of areolar and dense irregular connective tissue, right? That's the cutaneous membrane. Now, the skin chapter is right after this in your textbook, but I've moved it because I think you guys need more help with bones than you do with skin. So what I will do is I'll pick up skin a little later after I've helped you guys with bones and muscles, because that's really the hardest part of the first part of the semester. So we'll go back and pick it up a little later. I'm going to start on bones because you guys are going to start on bones this week, and I want to give you sort of a framework for studying bones. Cool? Oh, I think I said this already. Did I say it? Did I tell you guys you're having a quiz on Thursday? Okay, you are having a quiz, but it's only over histology. Okay, not bones. Okay, so has anybody gone to lab this week? Not yet. Okay, so this is the week, as I mentioned, in which people start feeling overwhelmed in lab, and it won't just be you. So if you feel like you have that deer in the headlights look in lab, realize at least that you're not alone, right? And then just go out and have a beer together afterward and talk about the foramen rotundum or whatever the hell you want to talk about. Okay. So find yourself a study group. I don't like studying with people because I'm an introvert and I don't really like people that much. But in this particular situation, study groups are actually really, really helpful in anatomy. I'm going to assess you guys mid-semester to see, have you guys self-report your grades anonymously and then ask a little bit about how you're studying to see if I can get another prediction about, like, let's see what A students are doing, right? Are they in study groups? Are they going to open lab? Do they use some techniques that I didn't know about? And then I'll spit the information back to you, okay? So before we get into the material specifically, let me tell you what I do with the skeletal system, what they do in lab, and then how they correlate, okay? So the skeletal system kind of covers a little bit of, well, it covers all of 3 chapters and then a little bit of the 4th chapter. It's chapter 6, 7, 8, and 9. Chapter 6 is an overview of the skeletal system, giving you sort of the logic of the system and how it develops, right? I do a lot of that in class. Chapter 7 is axial skeleton, the skull and all of that kind of stuff, which you guys will be doing in lab this week. Chapter 8 is appendicular skeleton. And then chapter 9 is articulations, which really is putting skeletons together with bones, ultimately. Chapter in class, and you do 7 and 8 in lab, but I use 7 and 8 and all of your examples from lab as reinforcing examples as I go through the entire time. And then here's something that's important for you guys to know. Whenever I put this at the top, pay attention to it, because all of the bones on your lab list you will be tested on in lecture, okay? I want you to study them at the same time. So why would I do that? Well, I can do that because instead of, for instance, teaching you what a condyle is, I can show you what a condyle is, and then I know that you learned the mandibular condyle on the mandible, and I can ask you about that one. In addition, I can make sure that you are studying lecture and lab together at the same time by giving you lecture quizzes that have a few of your lab bones that I know you're supposed to be studying. Now, I can't test the same way in lecture over bones as they can in lab, because you got a 3-dimensional structure, right? So I will promise you that although you are responsible for all of the bones that you learn in lab and the features, if you can hardly see it on a 3-dimensional structure, there's no way in hell I can ask you on a 2-dimensional picture in lab. But if you can't find a foramen magnum in any picture of the skull, then you should flunk it. Because it literally means big hole. Okay, it's a big hole, right? You can find the head of the femur, right? So the features that I ask you will be features that are readily identifiable on a 2-dimensional picture that would photocopy well or project well, right? Okay, so I will reinforce that. And what I do really is to help you with lab by forcing you to study lecture and lab at the same time and make them into one story. Okay, so it usually goes together relatively well. So you don't have a skeletal system quiz yet, though. Okay, so I want to briefly talk about the functions of the skeleton, and then the very first thing I'm going to do is try to get you guys ready and hooked up for lab by giving you some tips for studying labs successfully. First off, let's talk about the functions of the skeleton. Because of all of the collagen and of the solid nature of the matrix, it is supporting your body against gravity, right? Without it, you would be a flesh-colored pancake. It also is protective in some places. Some places, its primary function is protective. Can you think of any place that is less about any other function than protection? Where? Ribs and skull, yeah. The skull does have muscle attachments on it, but not a whole hell of a lot, honestly. And the ribs have some muscle attachments as well, but they're pretty protective for the squishy stuff underneath. Your primary job with the skeletal system that we'll really be talking about a lot this semester is that it provides leverage for body movement. Your skeleton doesn't move anything, but it provides leverage. So imagine that what I'm trying to do is I've got a rope, a tendon, and what it's attached to is something in the ground that I'm trying to pull out, right? So in order to pull out, what I'm going to have to do is I'm going to have leverage by standing here to pull on that. Same thing happens with your deltoid muscle right here. I use the clavicle and the scapula for leverage to move the humerus. Yeah? So your skeleton provides leverage for body movement. And it also does some other functions. It also stores calcium and phosphorus for you. It is your calcium bank. Whenever you take in more than enough calcium, you store it in your skeletal system because you don't just need it for your skeletal system. You will find out in physiology, if you don't already know, that calcium is super duper important for other body functions, and it has to be around and accessible instantaneously because you use it for nervous system transmission, you use calcium for muscle contraction, and you use it for blood clotting. All of those things are relatively important to do quickly. So you have to have a bank that you can always like make withdrawals from. And then in addition, your skeleton also produces all of your blood cells, right? And including your platelets, which aren't even really cells. So the red bone marrow produces all that. Okay, so let's just start talking briefly about studying bones. So I want to give you some tips for studying bones so that when you go into lab, you're less likely to feel overwhelmed. You are going to feel overwhelmed, but it'll be okay. And then you'll look back at it in 4 weeks and you'll be like, that was not that bad. This is bad. But then you'll get past that and you will be fine, right? Okay, so here's the deal. First thing is that there are a lot of names for bone features and bone markings. So here's some of them, okay? So you've got condyle, facet, head, alveolus, fossa, sulcus, all of those things. Now, you will, for instance, have, let's talk about that. This is figure 617 in your textbook. There's also, if you bought a lab book, there's also a list of them in your lab book, right? You would have a mandibular fossa and a mandibular condyle. And remember that with your lab practicals, they are not multiple choice. They are fill in the blank, which means that you have to be able to pull up fossa versus condyle, right? Fossa, condyle, foramen, right? Which means you need to know what the words mean. So take a minute and learn what the features mean. A fossa is a depression, yes? And a condyle is a projection that's shaped like a knuckle, okay? So then if I have a mandibular fossa and a mandibular condyle, I can say, okay, well, I think that the mandibular condyle sticks up and fits into the mandibular fossa, which is how you actually make this joint, the temporomandibular joint. Does that make sense? So there's a logic to it, okay? You need to know the difference between a fossa and a foramen. A foramen is a hole, but a fossa is a depression, right? So then when you come into something and it's the jugular blank and it's a hole, you're like, well, it's the jugular foramen because I know what foramen means, right? So you are going to need to take some time to make sure that you understand these. And then what I think is a useful thing is that after you find a condyle, you're like, yeah, that is a knuckle-shaped projection. Go find all the condyles so that you get a picture of what a condyle looks like in your head. Does that make sense? Okay, so learn the names of the features. And then the next recommendation, and this is one that most people just will not do until they flunk something, but damn, I wish they would. And the next one is to, once you find a feature, you have to pair it or parent it with the bones. So stick with me for a second. This feature right here is called the mastoid process. Mastoid as in shaped vaguely like a boob. And they name everything after boobs and nipples because it was all named by monks, right? And they're like, I think this is what a boob looks like. So like, this is as close as I get. So this is called the mastoid process, so just stick with me. And a process is anything that's projecting off of anything else. So if you look at it, you're like, okay, mastoid process, got it. Okay, so here's what people do. They label, they say this is the mastoid process, okay? But then they don't go any further because the mastoid process is part of a bone. And if you want to make sure that you know what you're talking about, you do not label it. It's right here behind your ear, by the way. It's right here, sticking out. You do not say mastoid process. You say mastoid process of the blank bone. It's the mastoid process of the temporal bone, but you could be more specific. Mastoid process of the what temporal bone? Left temporal bone. Why would you do that? Why would you force yourself to study that way? Because somebody could tag that structure and name this feature, or they could tag the same structure and say name this bone. Yes? So how do you know which features belong to which bones? you get into the habit of always parenting a feature with the bone. Parenting the feature with the bone, right? It's a really good habit because you never know which part of the freaking temporal bone, which is so complicated, right? In addition, if you're like me and you have trouble with 3D spatial imaging, if you keep naming parts of the temporal bone, it eventually gives you a 3D image of what the temporal bone looks like from the inside of the skull and the outside of the skull and all of it underneath of the skull, because you can access it from all of those points. Does that make sense? So instead of calling this the ramus, ramus means branch, we call it the ramus of the what? Mandible, exactly. And it's okay if you don't know the names of the bones yet, just trying to give you examples, right? So this is the head, but a lot of you guys now know that we should call it the head of the what? The head of the femur, exactly. And in a little bit, you'll be able to tell which femur it is. Right? You need a little bit of practice to do that. Does that make sense? So parenting bones will make you a much, much better student. So they call that parenting. So good would be if we labeled this the zygomatic process. Better would be zygomatic process of the right temporal bone, or this one's left temporal bone on that picture, left temporal bone. So then there's no way your lab instructor could trick you because you'd know which pieces belong to which bones. Okay, when you can tell left or right, which of course your skeleton has a lot of paired bones, not all of them, but a lot of them, you're responsible for left and right. There are going to be techniques for telling left and right, but let's go ahead and start with one of them. So you'll do left and rights on axial skeleton this week, but they're easy because well, with the exception of the clavicles. The clavicles are a little hard to do lefts and rights, but almost everything else is easy. And then the head's all put together, so if you can't tell left and right, I don't know what to tell you. But when you are getting to things like the femur or the humerus, there are ways that you do left and right that can be really helpful. So let's talk about them. So what I like to do with any bone that I'm going to have to side, which is no left and right from, is by the time I'm a little more experienced with the bone, I want to be able to put it down on a table or have a lab partner put it down on a table and be able to tell how the person that bone belonged to was facing without touching the bone. Does that make sense to you? So figure out where the head is and where the feet are and whether they're face up or face down, right? So you're trying to empathize with the person. Does that make sense? Okay, so I'm looking at this bone right here. This is the femur, and you don't have to know this material yet, but I'm going to give you like a way to organize it. So here's the deal. You put a femur down. Let's say I put it down on the table that way, okay? What you need to do in order to be very dependable about siding bones is always have an anterior or posterior landmark that you're very confident about. Okay, I don't care what it is. If you memorize an anterior landmark and you can't see it, you're looking at the back side, right? If you memorize a posterior landmark and you can, you're looking at the front side. So my anterior landmark is that if I put the femur down on the table, for instance, it makes a rainbow, not a smile. Because if you look at anybody's femur, if it had an arch to it, it would arch anteriorly, right? And that's true pretty much of almost all of our long bones, right? Okay. So the femur arches anterior. So if I put it down, it looks like a rainbow. I'm looking at the anterior surface, right? If I put it down, it looks like a smile. I'm looking at the posterior surface. Got that? You can use any one you want. It doesn't make any difference. And then you have to have a lateral and a medial landmark. Yeah? So what's a really good medial landmark for the femur? The head is a medial landmark, right? So now if I have it laid down, and let's say you are looking at the anterior surface, because you are looking at the anterior surface here. So you're looking at the anterior surface of the femur, right? And then I've got the medial landmark of the head. And so now I empathize with the person. Is this person face up or face down? Face up. Where's their head? Feet. Okay. And now can you tell me, based on that lateral medial landmark and the fact that we know anterior, posterior, is this a left or a right femur? Left. Left femur. Yes. Okay. So you need to get a process of elimination for siding bones, not just look at it and guess, right? I mean, you got a 50-50 shot, but I'd like a better statistical chance than that, wouldn't you? So you need to have a convention that you use. What anterior, posterior landmark you use is all up to you. Look at more than one bone, though, right? Get like 3 femurs and figure out what can I always tell on these 3 femurs, right? Okay, and then remember we have articulated skeletons too, and the articulated skeletons, if you ever are having trouble figuring out how it fits together, go look at it, right? Come back and look at the disarticulated one. So that's how I like to do it. For each bone, find an anterior, posterior landmark, lateral and medial, and that will help you side bones, especially when you get to long bones. And then go to the library or go to the STEM center or go to Open Lab and practice doing this. It's kind of a nuisance, but do it anyway. With a partner, unless you want to do it by yourself, pull out a bone, slap it on the table, don't touch it, figure out whether it's left or right. Please know that once you get to the lab practical, it's highly unlikely that you will be able to flip the bones around anyway. So most lab instructors do not let you touch the bones during the practicals and quizzes. And why? Because if I lay it down this way and then the other dude flips it over, he's got a different question than you, and also he might not be able to see what I labeled, right? So you need to get to the point that you can figure out, no matter how it's laid down, what it is, right? So don't just get comfortable that I'm always able to identify the features of the femur from an anterior view. And if possible, remember that you have a resource here, okay? You have a resource. You can feel the curvature of your clavicle. Now, once you have a clavicle in front of you, you feel the curvature of your clavicle, and you're like, oh, I can see how this goes to that. Nobody can take this away from you, and it ain't cheating, right? This is your lateral malleolus right here. This lateral malleolus I can feel going up to the bone that is lateral, and that is the fibula right there, lateral malleolus of the fibula, right? And then I can feel my anterior superior iliac spines right here. So on your hands, once you start learning the carpal bones, you can start to try to find them in your own carpals. And I know it seems silly to do all this stuff, but remember that when you're trying to learn a lot of stuff, the more senses you engage, the better the recall. So you can use your own body. You can feel the manubrium body and xiphoid process of your sternum, right? You can find your true ribs and your false floating ribs, right? You can feel them on your body. And you will look stupid, but no one is looking at you, I promise. They are not. They are trying to get through anatomy. That's all they are doing. And then last thing, you guys are a little ahead of the other class, so I'll let you go a couple minutes early. Each time you practice identifying a bone, get into the habit of, instead of just saying it, you need to write it. Because what are you going to have to do? You're going to have to write pterygoid process. And it's got to in it, right? And you are not going to do that well the first time on a quiz if you've never done it before, right? So write it. If you're practicing with a partner, check their spelling. Swap papers, check their spelling. Write it down over and over again. Okay, good luck. I hope you have fun in lab this week. I will see you guys on Thursday, histology quiz.
Thank you.
Uh-huh.